A very good afternoon to everyone. Welcome to today's class. And as we say in the beginning, that let's crack NEET PG together. So let's begin with the class. So today we are going to talk about vitamin B. Vitamin B. So we'll be covering all the types, that is 1, 2, 3, 6, 7, 9 and 12. We'll be talking about them. But before beginning with the class and the major topics, let us have a small introduction about Unacademy. So, Unacademy is one of the India's largest online platform for the preparation of NEET PG examination. India's best educators, they teach at Unacademy. So, you can get benefited of being taught by the best educators all over India for the preparation of the examination. Unacademy is very different from the normal online classes, and it is very special also, as you can ask your doubts directly with the faculty member, and they will be very much willing to solve your queries and doubts. So what is this special classes is they are live actually, and you can ask your doubts and questions directly with the faculty member. Then there are test series, and as I say that it is very important to know the pattern of the examination, to know the MCQs for qualifying any kind of examination. So that is possible with the help of test series. And these test series can be taken as number of times as much you want to. Once the quiz has been conducted, you can anytime go and go back to the quiz. Then this is one of the easiest way to prepare for the examination through online platform. It has got very flexible schedule. Then the language can be of your choice. Then unlimited practice is another added benefit. There are many faculty members who teach in the regional languages, so it is very much convenient way of preparation. Then you're not in town, or you cannot be present at that particular time over there in the class, then you can anytime just go and watch the video. So flexibility of schedule is the most important thing with the online learning. Then you won't feel anything different than a normal classroom, because you can chat with your educator. Then you can go for the interactive polls. There will be options, and you will be asked to take up one of the option, and then faculty members, they'll be explaining about each and every option to you. So interactive poll is quite interesting and interactive feature with an academy. Then you can learn anytime, anywhere. That this we have been saying since ever, right? Then there is this plus subscription, and what is so good about this plus subscription is that there are actually 19 subjects, and you can learn all these subjects with the help of plus platform. And for any upcoming exam, there is short-term course and crash course, which is like being conducted every now and then at an academy plus platform. So you can get benefited, and there are many students who have been actually benefited by the plus platform. So you can go for a 1 monthly subscription, 3 monthly, 6 monthly, 12 monthly, or 24 monthly. But the best is to go for a yearly or a 12 monthly subscription, because then you can make the most out of all of the data available at the Unacademy platform, right? So just go for this subscription and start preparation. And there is a certain charge which you need to pay for this plus subscription, that is like quite pocket friendly only. But still, you can get an additional 10% discount if you are using my referral code, that is FB01. So by using my referral code, that is FB01, you can avail an additional 10% discount. So don't forget to use my referral code, that is FB01, right? So yes, this was about the introduction of an academy. Let us begin with the topic, that is vitamin B. Vitamin B. Right? So I told you what all vitamins we are going to cover in this. And let us begin with the very first vitamin. So let's write that also with the pen only. That is vitamin B1, vitamin B1, and vitamin B1 is also called as thiamine. So first of all, we'll talk about thiamine. Yeah. So this is the first vitamin to be discovered in the vitamin B series, and that's why it has been given this one thing. So this was the first vitamin to be discovered in the vitamin B series, right? So let's write first vitamin to be discovered in vitamin B series, thus called as vitamin B1. So this is its name, and it is profoundly called as thiamine. Other than this, thiamine and vitamin B1, it is also called as anti-beriberi vitamin and anti-neurotic vitamin, right? So it is also called as anti-beriberi vitamin and plus anti-neurotic vitamin. So this is about the naming of vitamin B1, that is thiamine. Let's move further. That is, let's talk about the active form of vitamin. So active form of thiamine. So active form of thiamine is thiamine pyrophosphate. Thiamine pyrophosphate. What is the chemistry of thiamine? Talking about the chemistry, there is this pyrimidine ring. Then there is this thiazole ring. And the pyrimidine ring and thiazole ring, they are connected with each other with the help of methylene bridge. With the help of methylene bridge. And this whole thing together is called as thiamine pyrophosphate. TPP. Thiamine pyrophosphate. Uh, Sorry, this whole thing is called as thiamine. Whole thing is called as thiamine, and it is linked with 2-phosphate. And this 2-phosphate, when it is linked with thiamine, it gives rise to thiamine pyrophosphate. That is the active form of thiamine, right? Thiamine pyrophosphate. Let's highlight it. This thiamine pyrophosphate, right? So that is called as thiamine pyrophosphate, and which is the active form. So this is about the chemistry of thiamine. Now let us move towards another thing, that is the biochemical functions of thiamine. Biochemical functions. So as its active form is TPP, it serves as an important coenzyme. So as an important coenzyme, there are two important roles which are served by thiamine phosphate. So the two important roles which are played by thymine pyrophosphate is, first of the role is energy generation, and how exactly, that also we'll be seeing. And second role is transmission of nerve impulse. Transmission of nerve impulse. Right. Now, when talking about its further role, so it is required for this carbon-carbon bond cleavage. Requirement is in cleavage of carbon-carbon bond, required for cleavage of carbon-carbon bond. Now, if it is required for cleavage of carbon-carbon bond, where is it actually required? So, the first place where it is required for the cleavage of carbon-carbon bond is in the decarboxylation reaction of alpha keto acids. First is decarboxylation of alpha keto acids. Then second place where it is required is in the transketolase reaction of HMP shunt. Transketolase reaction of HMP shunt. So first of all, let us talk about the decarboxylation and then we'll talk about transketolase. So moving with the decarboxylation, let me just check whether my voice is clear. Yeah, so my voice is quite clear. Yeah. So moving with the decarboxylation. Moving with decarboxylation. So how is it helpful in decarboxylation? What happens is this pyruvate, when it is converted into acetyl-CoA, there is need of this TPP for the proper functioning of the enzyme, that is pyruvate dehydrogenase. Pyruvate dehydrogenase, it means TPP. There is removal of carbon dioxide and there is conversion of NAD into NADH plus hydrogen ion. Right? So this pyruvate dehydrogenase coupled reaction, it means TPP and it's get converted into acetyl-CoA. So this process is also called as oxidative decarboxylation. Oxidative decarboxylation. Right? And this PDH or pyruvate dehydrogenase, it is a multi-enzyme coupled complex. So multi-enzyme coupled complex. Right? So this was about decarboxylation reaction. Let us move and let us talk about the transketolase reaction. That where it is responsible for transketolase purpose. So what happens is this alpha ketoglutarate converted into succinyl-CoA in the presence of enzyme alpha ketoglutarate dehydrogenase, and there it means TPP. So this is how the alpha ketoglutarate is converted into succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase but it needs the support of thiamine pyrophosphate, right? Now the branch chain alpha ketone acid dehydrogenase, it is required over there also for valine, leucine and isoleucine. Right, so we were to discuss the two important reactions of thiamine pyrophosphate, that is decarboxylation and transketolase. And let us talk about some important other reactions which are catalyzed by thiamine. Some other reactions as well. So that is the transketolase reaction in HMP shunt. So that we have seen, and these are the two important reactions. Now talking about the sources. Talking about the sources of thiamine. So when we are talking about the dietary source, so unpolished rice is important source of thiamine. Unpolished rice. Unpolished rice is the important source of thiamine. So if the rice is polished, 80% of thiamine is removed, and 80% of thiamine is removed out because of this polishing. So this polishing is quite harmful for the thiamine. It can create thiamine deficiency. Then whole wheat, then yeast. So this was like about the vegetarian sources. Now talking about the non-vegetarian sources. So liver and kidney, specifically liver and kidney, they're rich in thiamine, right? So this was about the dietary sources of vitamin B1. Let us know that is talking about the RDA, that is recommended daily allowance of thiamine. It is 0.5 milligram per 1000 calorie intake, or it can be around 1 to 1.5 milligram per day, right? So this was about the recommended daily allowance of thiamine. Now talking about the deficiency manifestations. Deficiency manifestations. What are the deficiencies which are caused? So it is the major cause of beriberi. Beriberi. So what are the actual causes of this beriberi? Is because of the inadequate consumption of thiamine. Inadequate consumption of thiamine. Then excessive consumption of polished rice, if it is consumed in high amount, so there will be deficiency of thiamine. Then overcooking also loses thiamine. Overcooking can kill the thiamine. So overcooking. Then tea and coffee, it contains an enzyme that is called as thiaminase. Thiaminase. And it causes theoretical deficiency of thiamine. Only the theoretical deficiency. So some of the causes of beriberi are these, and let us talk about some other causes as well. So if there is chronic alcoholism, then also it causes beriberi. And whenever there is chronic alcoholism, there will be actually decreased absorption of thiamine in the body. So decreased absorption of thiamine. Decreased absorption of thiamine. Right? Now, if it is decreased absorption of thiamine, this is one thing, and there will be lesser conversion of thiamine into thiamine pyrophosphate, TPP, in chronic alcoholism. Then Uh, then diuretic therapy. Diuretic therapy is also cause of thiamine deficiency, right? So these are some of the causes of beriberi that we have seen: chronic alcoholism, diuretic therapy, then adequate consumption of vitamin B1, polished rice, overeating, then overcooking. These are some of the causes. Now, what are the symptoms of beriberi? So, what are the symptoms of beriberi? So, weakness is the very first symptom. Now, there are different types of beriberi also. So, types of beriberis. Types of beriberi. So, the very first type is dry beriberi. Dry berry. What happens in this is there is weakness, then there is peripheral neuropathy. Peripheral neuropathy. Right. So this was about the first type of beriberi, that is dry beriberi. Let us move towards the second type of beriberi, that is the wet beriberi. Wet berry. Now what happens in this is there will be severe manifestations. The symptoms will be quite severe. There will be breathlessness, then there will be accumulation of fluid in the pericardial space, and there will be edema. So fluid built up, and there will be edema. Then other things that will be seen in wet beriberi is cardiomyopathies. Then there will be dilation of heart. Dilation of heart. So this was about the wet beriberi. Then talking about the third type of beriberi, that is infantile beriberi. Infantile beriberi. So what happens is in this is there will be severe manifestations again. There will be acute fulmination of cardiovascular symptoms, and that is also called as Shoshin beriberi. Also called as Sho Shoshin beriberi. So the symptoms will be quite severe, and there will be cardiovascular symptoms. Then the fifth type is polyneuritis. Then sixth type is it occurs in the chronic alcoholism, and it is also called as Vernix Korsakoff syndrome. Vernix Korsakoff syndrome. So what actually this is like club of two things, that is Vernix and Korsakoff. So what are the symptoms of Vernix and what are the symptoms of Korsakoff? Just a second. Just a second. Yeah. So we were at Vernix-Korsakoff syndrome. So what happens in Vernix is there will be global confusion. The symptoms are global confusion and ophthalmoplegia and ataxia. Then in Korsakoff, what will occur? There will be memory loss and there will be psychosis. So the clubbing of the two things called as Vernix-Korsakoff syndrome. So the reason of thiamine deficiency in alcoholics is because of the decreased absorption of thiamine from the intestine, and if there is decreased absorption of thiamine in the body, it will also hamper the energy metabolism. That is, there will be decreased formation of ATP, and heart and brain, they require lots of ATP. Thus, the most affected tissue in Beriberi is heart and brain, right? So in alcoholism, that is because of the decreased absorption we have seen. Now in treatment, what would be given? So 100 milligram of thiamine per day for 7 days. For 7 days, and 10 milligram per day until complete recovery. Until complete recovery. So this is the part of treatment of thiamine, right? Then what about the diagnosis? Diagnosis. So there will be decreased transketolase activity in RBC in very. So what will we say? That there is a decrease in RBC transketolase activity. Transketolase activity in beriberi. And the level of thiamine and TPP can be measured by HPLC. So level of thiamine and thiamine pyrophosphate can be measured by HPLC. Right. So this was about thiamine. Let us move ahead with the second vitamin, that is vitamin B2, and that is riboflavin. So, vitamin B2, riboflavin. So this is also called as Warburg yellow enzyme of cellular respiration. Warburg, also called as Warburg yellow enzyme of cellular respiration. Then it can be also used as food coloring agent. Can be used as food coloring agent. So the color it provides is from yellow to orange. So it can be very safestly can be used as a food coloring agent whenever we need yellow to orange color in some food preparations, right? Then what another important thing about vitamin B2 is it is heat stable and it is light sensitive. It is light sensitive. Talking about the chemistry of riboflavin, so how it is actually dimethyl alloxane ring, that is dimethyl isoalloxazine ring, which is connected to ribitol, by a nitrogen, and this together is called as riboflavin. This together is called as riboflavin. So in chemistry, it is dimethyl isoalloxazine ring connected to ribitol, and this together gives us the riboflavin. And what about ribitol? It's a derivative of ribose, and instead of aldehydic group, it is replaced by the alcoholic group, right? So it's a derivative of ribose, derivative of ribose. And how? The aldehydic group is replaced by alcoholic group, right? Now, its active form is actually flavin mononucleotide, Fmn and Fad. These two are the active forms of riboflavin. So when we are talking about Fmn, it is actually riboflavin plus riboflavin with one phosphate group, and it is together called as flavin mononucleotide. Flavin mononucleotide. And if we are saying Fad, that means Fmn plus 1 AMP, and it is called as flavin adenosine dinucleotide, right? Here it is mononucleotide, here it is flavin adenosine dinucleotide, right? One from here and one from here. So these two, Fmn and Fad, are the two active forms of riboflavin, right? So these active forms, they are used as coenzymes, and they are the coenzymes which are used for various reactions, and they are tightly bonded. So let us write also. These two coenzymes, they are bonded tightly with the enzyme. So they are called as flavoproteins. The proteins along with the riboflavin is called as flavoproteins. Flavoproteins. So what are exactly flavoproteins? These are the tightly, Fmn and Fad, they are tightly bonded with some of the proteins, and these proteins are called as flavoproteins, and this attachment is actually non-covalent. The attachment is non-covalent. Attachment of Fmn and Fad is non-covalent, and that is why they are called as flavoproteins, right? So let us have some of the examples of flavoproteins. So examples of flavoproteins. What are the examples of flavoproteins? So first example is succinate dehydrogenase. Then second example is glutathione reductase. Glutathione reductase. Right. So we have seen about its chemistry. We have seen about some of the important functions or its active form in which it is used. Then let us talk about the biochemical functions. Biochemical functions. Biochemical functions of riboflavin. Right. So the first one is, it is also called as energy generating vitamin. Energy generating vitamin. and it is involved in cellular respiration and in redox reaction. Most importantly, in cellular respiration, it is responsible for the various redox reactions, and this nitrogen of alloxazine ring is actually responsible is for redox reactions. For redox reactions, this N that we have seen in the structure, that how dimethyl alloxazine, isoalloxazine is attached with ribitol, this N. So this is the most important thing which is responsible for all type of redox reaction functions of our riboflavin, right? So this FAD and Fmn accept the nitrogen and get converted, accept the hydrogen and they get converted into FADH2 and FmNH2. So these are the type of reactions will get themselves reduced. So reactions which are catalyzed by Fmn, first of all, reactions catalyzed by Fmn, reactions catalyzed by Fmn. So Fmn is actually used as a prosthetic group or as a coenzyme. So it is used as prosthetic group or as a coenzyme, right? So the reactions which it catalyzes is L-amino acid oxidase, the enzymes in which it is involved, then monoamine oxidase, then it is involved in the complex 1 of electron transport chain. Complex 1 of electron transport chain through NADH dehydrogenase. NADH dehydrogenase. NADH dehydrogenase. So the reactions which are catalyzed by Fmn are actually reactions which are based on L-amino acid oxidase, monoamine oxidase, and complex 1 of the electron transport chain, that is through NADH dehydrogenase. So this was about Fmn, and let us move and talk about FAD, the reactions which are catalyzed by FAD. So the reactions which are catalyzed by FAD, what happens is this FAD, it accepts the hydrogen and itself gets converted into FADH2, and one FADH2 releases 1.5 ATP. Right. So let us talk about the enzymes where it is required. So first of all is succinate dehydrogenase. Succinate dehydrogenase of complex 2 of electron transport chain. Then it is responsible for acyl coenzyme dehydrogenase, acyl coenzyme dehydrogenase. Then it is required for d-amino acid oxidase. Then it is required for xanthine oxidase. Then it is required in glutathione reductase. And this glutathione reductase is responsible for the antioxidant activity. So it can be called as a kind of antioxidant vitamin. Then there are two more enzymes, that is pyruvate dehydrogenase, pyruvate dehydrogenase, and alpha ketoglutarate, which we have seen, alpha ketoglutarate in vitamin B uh, dehydrogenase. So these are actually multi-enzyme complexes, and they require some five other coenzymes also along with the vitamin B2. It also requires thiamine, pyrophosphate, it requires FAD, it requires NAD, it requires lipoic acid, then it requires coenzyme A. Coenzyme A. So it is a group of multi-enzyme complex, and it requires total five of the different important coenzymes. Right, now talking about the sources of riboflavin, that what are the sources of riboflavin. So talking about the non-vegetarian sources are liver and kidney. Then in dairy it is present in milk. Then cereals, the outer layer of the cereals. Right, so this is the sources. Now talking about RDA, recommended daily allowance of riboflavin, that is 1.5 milligram per day. 1.5 milligram per day. Now talking about the deficiency manifestations. Deficiency manifestations. What are the deficiency manifestations? So when talking about riboflavin, there is actually no as such deficiency, but it can occur as a part of multivitamin deficiency or protein energy malnutrition, that is the PEM. So there is no as such particular deficiency which is related. Now in chronic alcoholism or in several alcoholism, there is a condition which is called as erythrobioflavinosis, that is erythrobioflavinosis. What happens in erythrobioflavinosis is, let us talk about the symptoms. Let's just highlight this and move towards the symptom. Erythrobioflavinosis. So what are the symptoms of erythrobioflavinosis is, there will be glossitis. That is, the tongue will become purple or red, and it is also called as magenta tongue. Magenta tongue. Magenta tongue. And there will be loss of capillary action of the tongue in glossitis. Then second is chiliosis, that is cracking of the side of the mouth will occur. Then angular stomatitis. Then next thing is seborrheic dermatitis. Then there will be corneal vascularization. So these are some of the symptoms which are associated with erythrobioflavinosis. Right. So talking about the lab diagnosis of deficiency of vitamin B2 or riboflavin, that it is done by measuring the RBC, glutathione reductase activity. measuring RBC, glutathione, reductase activity, reductase activity, about the lab diagnosis of riboflavin. So like this, we have done our second vitamin of the day, that is vitamin B2 or riboflavin. Let us move towards the third vitamin, that is vitamin B3 or niacin. Vitamin B3 or niacin. So talking about vitamin B3, it is also called as nicotinic acid. Nicotinic acid. And the amide form of this nicotinic acid is called as nicotinamide. And it is also called as niacinamide. Niacinamide. Right? Now, talking about the chemistry of niacin, what happens is it is made up of pyridine 3-carboxylic acid. Pyridine 3-carboxylic acid. And it is not a true vitamin. It's not a true vitamin. Why? Because there is partial synthesis of niacin from tryptophan. So it's not a true vitamin, right? So how much amount of niacin is produced from tryptophan? So let us see this. Tryptophan, niacin. And while conversion of tryptophan into niacin, there is need of vitamin B2, there is need of vitamin B6, and there is need of iron. So 60 gram of 60 milligram of tryptophan will lead into formation of 1 milligram of niacin. So there is partial synthesis of this your niacin from tryptophan. Now talking about the structure of this pyridine 3-carboxylic acid, it actually looks like this. Here there will be one N and this is CO NH2 CO1H. So this is how the structure of niacin looks like. Now, the active form of niacin or the coenzyme form of niacin, like for every vitamin, we are talking about the active form of the particular vitamin. So the active form or the coenzyme form of the vitamin B3 or niacin is your NAD positive and NADP positive. So coenzyme of niacin. So they are NAD positive and NADP positive. So they are responsible for generation of 2.5 ATP. And when we were talking about FAD, was responsible for generation of 1.5 ATP. Here it can give 2.5 ATP, and it is always represented by this positive sign. You can see this positive sign. So why is it that it is always written with this highlighted plus? Why? Because the nitrogen is in quaternary form, is in quaternary state, and thus it is represented as N positive, and thus represented as N positive, right? So if you are saying that the two active forms are NAD and NADP, so what are the full forms of NAD and NADP? So NAD is nicotinamide, adenine, dinucleotide, right? And NADP is nicotinamide, adenine, dinucleotide phosphate. So these are the full forms of NAD and NADP, right? When we have talked about the coenzyme form and active form of the enzyme vitamin, let us move towards the biological function of NAD and NADP. So biological function. So they are actually involved in various types of redox reactions and they accept the hydride ion. So the very first and the most important is involved in redox reactions and accept the hydride ion. So the reactions which are actually catalyzed by NAD, the reactions which are catalyzed by NAD as a coenzyme. So it is highly important in glycolysis and Krebs cycle. So I'll be mentioning the names of the enzyme where this NAD is used. So in enzyme glyceraldehyde 3-phosphate dehydrogenase NAD is required. Then lactate dehydrogenase it is required. Then pyruvate dehydrogenase it is required. Then isocitrate dehydrogenase it is required. Then alpha ketoglutarate dehydrogenase it is required. Then malate dehydrogenase it is required. Then in beta hydroxy acyl OA dehydrogenase it is needed, and it is also needed in beta chain keto alpha dehydrogenase enzyme. So NAD is required at all these places. And when we are talking about NADP, so let us talk about the places where it is needed. So it is needed in the malic enzyme and it is needed for phosphogluconate dehydrogenase, and it is also needed for glucose 6-phosphate dehydrogenase. So these are the places where NAD and NADP is used as coenzyme, right? This uh, active form of niacin or our vitamin B3. Now, talking about NADP only, continuing this thing that it is required for various reductive biosynthesis, required for reductive biosynthesis. So it is required for fatty acid synthesis. It is required for vitamin D synthesis. It is required for cholesterol synthesis. It is required for the synthesis of steroid hormones. Required for synthesis of steroid hormones. So this is how the uses of NADP and NAD as biological functions. Now talking about NAD and NADP, this NAD and NADP, they are also required for ADP ribose supply for ADP ribosylation of protein and DNA repair mechanism. So they are highly important in ADP ribosylation. ADP ribosylation reaction for various DNA repair mechanisms and etc. Now talking about the sources of niacin. What are the sources of niacin? So the very good sources are non-vegetarian sources, that is liver, kidney, fish, then cereals are very good sources of vitamin B3. And the deficiency of niacin occurs in jowar and maize eating population. Jowar and maize eating population suffer from its deficiency. Right. So we have seen the sources and where it is actually it causes deficiency that we have seen. Then talking about the RDA, recommended daily allowance of vitamin B3, that is 20 milligram per day. 20 milligram per day. Right now, moving towards the deficiency manifestations. Deficiency manifestations. So, diarrhea, then dementia, then dermatitis, and most importantly, pellagra. So, 3D dermatitis is classical dermatitis, the skin which is exposed in sun. It gets a very different type of appearance, that is around the neck. It is called as castle's necklace. And if it is on the hand and feet, it also gives appearance like gloves. So gloves or stocking like appearance it gives. So deficiency manifestation is diarrhea, then dementia, and then dermatitis, right? And if it is left untreated, it might cause death. Untreated might cause death, right? So when does this deficiency it occurs? Deficiency occurs whenever a patient is on treatment of tuberculosis. If a patient is on treatment of tuberculosis and he's given with isoniazid hydroxide, and this inhibits the PLP. It inhibits PLP, and this PLP is actually required for kynurenine pathway or activity in which there is conversion of tryptophan into niacin. So this deficiency of PLP is related to tryptophan metabolism and which is indirectly linked with synthesis of niacin. So whenever there will be deficiency of PLP, it will actually cause pellagra-like symptoms. Pellagra-like symptoms. Right. So coming to another disorder related to this is Hartnupp's disease. Hartnupp's disease. So there will be defective absorption of tryptophan itself from the intestine, and in this also there will be pellagra-like symptoms. So defective absorption of tryptophan from intestine. And the another disease that is related is carcinoid syndrome. Carcinoid syndrome. So the deficiency of tryptophan, because it is diverted towards serotonin. Thus, there will be again pellagra-like symptoms, right? So deficiency of tryptophan because diverted towards serotonin, and it causes again pellagra-like symptoms, right? Then If talking about when is it given and treatment of which all thing this niacin is given. So niacin is given in uh, treating the hyperlipidemia. Hyperlipidemia. So this helps in reduction of the LDL level. It helps in reduction of triglyceride levels, and it also improves the HDL level. So it is given in the various actually lipid related disorders. Niacin is given, and the two things that we have seen which is linked to niacin is heart nerve disease and carcinoid syndrome. Now this was about the niacin. Let's move towards our next vitamin, that is vitamin B6. Vitamin B6, and it is also called as pyridoxine. Pyridoxine. So talking about this, so it has got one active form or one coenzyme, and we'll be discussing it further. But before that, we'll just move towards the recommended daily allowance of pyridoxine. So the recommended daily allowance is 2 milligram per day. And uh, what about its structure? That how it is looked? So it is a pyrimidine derivative. So pyrimidine derivative. It's a pyrimidine derivative. So how does it look like? And here there will be one CH3. So here, as you can see, this is alcoholic group, right? Here, if you can see, this is alcoholic group. So if it is alcoholic group, it is called as pyridoxine, right? This is a normal pyridoxine. Pyridoxine. If this alcoholic group is replaced by aldehydic group, it is called as pyridoxal. And if it is replaced by a amide, it is replaced by a amide, then it is called as pyridoxamine. Pyridoxamine, right? So this is about its structure or the chemistry, pyridoxine. Now, when we were just discussing in the starting that there is one active form of pyridoxine, there is one coenzyme of this pyridoxine, and this one active form is called as PLP. So what is the full form of PLP? That is pyridoxal phosphate. Pyridoxal phosphate. So this we have seen. Then we always talk about the source of particular vitamin. So the source is, yes, Vijay Kumar Palla. PLP is pyridoxal phosphate. So sources is fish, milk, meat, wheat, rice, and etc. Right? Then talking about the biochemical functions. Biochemical functions. So first of all, it is required in transamination reactions. It's required in the various transamination reactions. So transamination reaction. So In transamination reaction, it serves as a coenzyme, right? So one of the example of this reaction is there will be conversion of amino acid into keto acid, and the thing will be this NH2 will be converted into pyridoxal phosphate, and it will be further converted into pyridoxamine phosphate. And okay, let us draw a much clearer picture. I'll just do it from the very starting because then we had to change this slide. So this amino acid is getting converted into keto acid, and the PLP will get converted because of there will be addition of the amine group, will be converted into pyridoxamine phosphate. Pyridoxamine phosphate, and this pyridoxamine phosphate will be given to another keto acid, and this keto acid will enter the TCA cycle and it will give energy. And it might also get converted in another amino acid. Anything is possible. So what can be said by seeing all this, that pyridoxine is an important vitamin required for energy generation required for energy generation, right? And what another thing is associated with pyridoxine, that it is also required for hematopoiesis. So this we'll be seeing further that how is it needed. Here we have seen that how it is involved in the transamination reaction, and in transamination reaction how it is further useful in generation of energy. Right. So let us talk about the another reaction. This is the transamination reaction, the first biological function. Now let us talk about the decarboxylation reaction. It is involved in the decarboxylation reactions also. So how is it helpful in decarboxylation reactions? There will be formation of neurotransmitter, that is 5-hydroxytryptophan. The neurotransmitter 5-hydroxytryptophan, PLP will be needed, and there will be decarboxylation and it will get converted into serotonin, that is a neurotransmitter, and DOPA is again by the help of PLP and by decarboxylation it will get converted into dopamine, and this dopamine is a neurotransmitter, right? This is neurotransmitter, that is further it will be responsible for non-epinephrine and epinephrine, right? Then the decarboxylation reaction, glutamate, it will be converted into GABA with the help of PLP and removal of carbon dioxide. So what is the full form of GABA? That is gamma amino butyric acid. So whenever there will be deficiency of neurotransmitters, when we are talking about the PLP and it is linked with the formation of neurotransmitter, so whenever there will be deficiency of neurotransmitter, it will cause peripheral neuropathy. It will also cause convulsions. It will cause depression, etc. Right. So we have seen two reactions, that is transamination and then decarboxylation. Now, the third important reaction, which is actually done with the help of PLP, is transsulfuration reaction. Transsulfuration reaction. So what happens in this is there will be conversion of methionine into cysteine. What happens is the enzyme cystathione beta synthase. Cystathione beta synthase is involved and it actually needs PLP. So whenever there will be deficiency of this cystathione beta synthase, it causes a disorder that is called as homocystinuria type 1. Homo deficiency of cystathione beta synthase, it causes homocystinuria type 1. And it also leads to various cardiovascular manifestations. So this PLP is actually required for hundreds of reactions of amino acid metabolism. Hundreds of reactions in the metabolism of amino acid. The another reaction which is catalyzed by PLP is heme synthesis. So what happens in heme synthesis is there will be, let us take another slide. There will be condensation reaction between succinyl OA and glycine. This is a kind of condensation reaction and it will get converted into delta ALA. So it requires ALA synthase and this ALA synthase is actually PLP dependent. So whenever there will be deficiency of PLP, here in this case, whenever there will be deficiency of PLP, it causes sideroblastic anemia. It causes sideroblastic anemia. Right? So this was about how it is required in heme synthesis. Then it is also required for conversion of tryptophan into niacin. Right? So it is required for the synthesis of niacin from tryptophan. This we have just seen in the last reaction, or in the last vitamin. We have seen how niacin is formed from tryptophan. So in tryptophan metabolism, let us talk that this tryptophan is converted into 3-hydroxykineurenin, and this 3-hydroxykineurenin is converted into 3-hydroxyantranilate. It's converted into 3-hydroxyantranilate, and this conversion of 3-hydroxykineurenin into 3-hydroxyantranilate is with the help of enzyme that is kineurinase. Kineurinase, and this kineurinase is PLP dependent. Yes, kineurinase requires PLP. Vijay Kumar Palla. So whenever there will be deficiency of PLP, wherever there will be deficiency of PLP, what will happen that xanthine urinate will be excreted out in urine. Right? So this is another thing related with your PLP. Then it is also required for glycogen phosphorylase function. It is required for glycogen phosphorylase function. That is, it is required for glycogen metabolism as well. So how? This glycogen is converted into glucose with the help of glycogen phosphorylase, and this glycogen phosphorylase is PLP dependent. This is PLP dependent. So we have seen all the biochemical reactions which are related with the pyridoxal phosphate, and let us talk about the deficiency manifestations before moving towards the next enzyme. So let us talk about the deficiency manifestations. Deficiency manifestations. So the first one is peripheral neuropathy. Peripheral neuropathy. And this is because of the decreased serotonin and dopamine. Peripheral neuropathy is because of decreased serotonin and decreased dopamine. Then the second deficiency manifestation is convulsions. So this is because of the decreased GABA, that is gamma butyric acid. Decreased GABA will cause convulsions. Then it will also cause pellagra-like symptoms. Pellagra-like symptoms. So pellagra is actually because of the deficiency of niacin, but the similar kind of symptoms will be seen here also in the deficiency of PLP, that is in pyridoxine. Also, the pellagra-like symptoms will be manifested. Then fourthly, xanthurinate will be excreted out in urine if there is defective tryptophan metabolism. So xanthurinate in urine. Then the patient will suffer from anemia as well. Right, so these are some of the deficiency manifestation. Now talking about the drug interactions. Drugs interactions. So first of all, in INH, that is in tuberculosis treatment, it will interfere. Then in penicillamine, penicillamine. So this is given in tuberculosis. This is given in Wilson's disease treatment. It will also interfere. Then cycloserine, and then L-DOPA, which is given in Parkinson's disease. So these are some of the drugs which actually affect the level of pyridoxine, So these are the drugs which are quite interfering in the pyridoxine. So these drugs cannot be given all alone. They cannot be given all alone, and they must be given with pyridoxine. Otherwise, they will cause pyridoxine deficiency in the body. So they are always given with pyridoxine. Now, let us see the diseases in which this is used. This pyridoxine is used. In which diseases it is used. So, important diseases in which is used. So the diseases in which it is used is homocystinemia, then in anemia, then in retinal problems like atrophy is given. Then mega doses of pyridoxine is also given in certain cases. So mega dose is also given in PMS, premenstrual symptoms, then in schizophrenia, then in carpal tunnel syndrome, So always remember the mega dose is given where? Given in PMS, schizophrenia and carpal tunnel syndrome. So these are some of the important diseases in which it is used. Now, let us talk about the diseases which are caused because of the deficiency of pyridoxine. So, diseases because of its deficiency. Diseases because of its deficiency. So, first is pellagra-like symptoms, then oxaluria, then homocystinemia, then homocystinuria, then the another thing is various cardiovascular diseases it will cause, and it will also cause the excretion of xanthiurinate in urine. Xanthiurinate in urine. Right. Now, so vitamin B6, it is a commonly prescribed drug. Let's talk about it here by another color. So yes, in morning sickness and pregnancy also it is given with along with the other drugs. So in morning sickness, in pregnancy, along with other drug, it is used, and it is also given for diabetic neuropathy. Also given for diabetic neuropathy. Right. So this we have learned almost everything about pyridoxine, that is your vitamin B6. Now talking about another vitamin, that is vitamin B7, and that is biotin. So vitamin B7 or biotin. So this is also called as vitamin H, also called as vitamin H or coenzyme R. Coenzyme R. Then talking about the sources, so it is present in milk, it is present in egg yolk, present in egg yolk. Then the egg white, it contains avidin, and this avidin is called as antidote for biotin. Antidote for biotin. So biotin is also called as anti-egg white injury factor. This biotin, also called as, yes, Vijay Kumar Palla, yes, it is also called as anti-egg white injury factor. Okay, so this was the first thing, that where it is present. Then biotin is widely used in beauty industries. Biotin is highly used in all of the products which are for the hair treatment. So it is present in shampoos, then it is present in creams. It is used for various hair loss problems. But one thing should be made clear that it has got very little absorption from skin, very little absorption from skin. So no matter how much hard one tries to, its absorption is less from skin. Then another thing is this biotin, it actually works in association with ATP, and usually wherever it is required, it is in association with ATP and carboxylase. So in all of the carboxylation reaction, you will always find a combination of ATP, biotin and carboxylase. So whenever carboxylation is occurring, it will also be with the help of biotin and it will be related to ATP, right? So let us talk about the biotin dependent reactions. So biotin is required in certain reactions. So talking about the biotin dependent reactions, we'll be talking about certain reactions, that is around 3 to 4 reactions which are biotin dependent. So biotin dependent reactions. So biotin is not required in certain carboxylation reactions, and in certain of them it is required, right? So it is required in CO2 fixation of the C6 of purine. So C6 of purine, CO2 fixation of the sixth carbon of the purine ring, then it is required for conversion of pyruvate into malate. Here this malic enzyme will require biotin. Then the third place where it is required is it is required for gamma carboxylation in vitamin K. So these are some of the places where it is required. So now talking about its chemistry. And talking about its structure also. The structure is like this. The structure is like this. There will be one N. There will be one N again. And this ring is called as imidazole ring. Imidazole ring. And this place, here it will contain one sulfur. And here it will contain valeric acid, which is linked with the carboxylic acid, and this ring is also called as thiophene ring. And this nitrogen is responsible for carrying carbon dioxide carrying capacity this nitrogen will have, right? And the lysine enzyme will act over here. So this is how your biotin looks like. Let us move further. Let us talk about the sources where it is obtained from. So sources, we have just seen that it is present in good amount in egg yolk. So it is present in yolk of egg. It is present in meat, fish, milk, tomatoes. Then it is present in grains, etc. And it is also synthesized by intestinal flora. So its deficiency is a very rare thing because it is self-synthesized in the body. Then talking about RDA, that is the recommended daily allowance, that is around 30 to 100 microgram per day. It is required. Then moving towards is biochemical functions. Biochemical functions. So it is important in gluconeogenesis reactions. Important in gluconeogenesis. So seeing this further, that is conversion of pyruvate into oxaloacetate. There will be need of carbon dioxide, there will be need of ATP, and there will be need of biotin, and the enzyme which will be catalyzing this reaction is pyruvate carboxylase. Pyruvate carboxylase, and it is important for generation of energy from the non-carbohydrate sources. Important for generation of energy from non-carb sources. Non-carb sources. Right. So the first thing, then it is also required for fatty acid synthesis. Required for fatty acid synthesis. Fatty acid synthesis. So how? The acetyl-CoA will be converted into malonyl-CoA, and it will require acetyl-CoA carboxylase. Acetyl-CoA carboxylase, and again it will need A, B and C. This A is for ATP, B is for biotin, and C is for carbon dioxide. So it is required for fatty acid synthesis as well. So the two reactions where it is required, we have seen, that is conversion of pyruvate into oxaloacetate, that is for the generation of energy. Then for fatty acid synthesis. Now let's move further, that is in the leucine metabolism. Required for leucine metabolism. Leucine metabolism. Then the fourth place where it is required is the place where there is conversion of propionyl CoA into methyl malonate CoA and then succinyl CoA. That is conversion of propionyl, that is odd chain fatty acid propionyl CoA into methyl malonyl methylmalonate CoA into succinyl CoA, and the enzyme required here is propionyl CoA carboxylase, and it will again need the ABC, ATP, biotin and carboxylase. So this conversion is important because this is for the odd chain fatty acid, which is converted into further and goes into TCA cycle. This goes further into TCA cycle and it is completely oxidized. It's completely oxidized, right? So it is a kind of biotin dependent carboxylation. It is biotin dependent carboxylation reaction, right? So let us talk about its deficiency manifestations. Deficiency manifestations. What are the deficiency manifestations? So as such, there will be no deficiency because it is produced in the body. The intestinal flora itself produces it, so there will be as such no deficiency. And because it is also abundantly present in the food, almost every food it is found, so again its deficiency is rare. But deficiency might occur in certain cases, that is when there is increased use of increased use of antibiotics, increased use of antibiotics, right? And whenever there is a prolonged antibiotic therapy, this will be occurring. Then neurological manifestations will be there. The neurological manifestations are hallucinations, then depression, then skin problems such as rash around mouth, face, and nose. So, skin problems in terms of rashes, and it will also lead to hair loss. So, many people they recommend biotin supplementation also in various hair loss treatments. Now, talking about the antidotes of biotin, that from which of the things it will be inhibited. So, let's take another slide and antidotes of biotin. So, from the very starting of biotin, we have seen the first antidote, that is avidin, right? We have seen avidin. So, avidin has a more complementary binding capacity with biotin rather than with antigen antibody complex. So, avidin is highly complementary to biotin as compared to antigen antibody, right? It will be binding with biotin as compared to antigen antibody, and that is why this avidin is highly used in ELISA test. It is used in DNA labeling. Is used in DNA labeling. Then it is used as DNA probes. So this is about avidin. Yeah. One more thing. There is one more form of avidin, that is streptavidin. Streptavidin. So this is also a form of avidin. Now, what is the use of biotin? Uses. So it is used for regulation of gene expression. Used for regulation of gene expression. So how does it regulate the gene expression? Via biotinylation of the histone proteins. The histone proteins, they are biotinylated, and thus it is also used for regulation of the gene expression, right? So we have seen about biotin also. Uh, Let us move towards our next vitamin, that is vitamin B9 or folic acid. So vitamin B9 or folic acid. So folic acid is actually water soluble vitamin, right? And it is responsible for RBC maturation. Responsible for RBC maturation. Then it is also called as, when it is responsible for RBC maturation, so it is also called as hematopoietic vitamin. Hematopoietic vitamin, right? Moving towards the chemistry and structure. So chemistry and structure. So there is a pteridine ring, pteridine ring, and it is joined with PABA, that is para-amino benzoic acid, and they together called as stearic acid, and this PAPA is attached with a glutamate, and this whole thing together is called as folic acid. Called as folic acid, right? So from where this folic word is coming from, it comes from a Latin word that is folium, that is for the leafy greeny vegetable. So it's a Latin word, and Latin word came from there because the sources of folic acid is actually green leafy vegetables. Green leafy vegetables. And that's why it is named after them. Now, as we are seeing some of the active forms of all of the vitamins, so let us talk about the active form of folic acid. Active form of folic acid. So this folic acid gets converted into dihydrofolate in the presence of NADPH dehydrogenase and NADPH dehydrogenase folate reductase, or it is also called as DHFR, right? And this dihydrofolate will get converted into tetrahydrofolate in the presence of DHFR and NADPH. And this tetrahydrofolate is a coenzyme, and it is responsible for various biochemical reactions. Right? So this is how the green leafy vegetables folic acid is converted into the active form. So let us move towards the next thing, that is the biochemical functions, right? So biochemical functions. What are the biochemical functions? So most importantly, it is involved in one carbon metabolism. So involved in one carbon metabolism. Now, how is it responsible for one carbon metabolism? There will be transfer of one carbon compound from one to another. So, compound from which it is transferable carbon is moved, right? So, transfer of one carbon compound means a compound which contains transferable carbon. So, their metabolism. So, the transferable carbon is present in ethene, then it is present in methylene, which is connected to tetrahydrofolate N to form a bridge between N5 and N10 of THF. So it forms a bridge between N5 and N10 of tetrahydrofolate. Then it is responsible for transfer of methynyl group. Methynyl group methynyl group, and it also forms bridge between N5 and N10, N5 and N9 of THF. N5 and of THF. Right. So then it is also required for transfer of formal group. Then it is responsible for transfer of hydroxymethyl group, hydroxy ethyl group. Then it is responsible for transfer of formamino group. Formamino group. Transfer of formamino group. Right. So in one carbon metabolism, the entry point is actually methylene tetrahydrofolate, and which gets converted into methynyl tetrahydrofolate. So let us write that in one carbon metabolism, the entry point is methylene tetrahydrofolate, which gets converted into methynyl tetrahydrofolate, right? And which get reversibly converted into formyl tetrahydrofolate, and which is important for this formyl tetrahydrofolate is actually responsible for the uh, C2 and C8 ring of purine. C2 and C8 ring C2 and C8 of purine ring, right? So this is the entry point. Then coming to the sources for one carbon compound, the sources for one carbon compound is serine, then it is glycine and choline will be methylene tetrahydrofolate. So this is the source for the one carbon compound in tetrahydrofolate. So this, it is needed for serine, it is needed for formamine. Now let us just move further in this whole thing, that how the deficiency of certain thing will was issued. So we have seen that methylene, this methylene tetrahydrofolate, it's converted into methylene methylene tetrahydrofolate. This we have seen. So this methylene tetrahydrofolate itself gets converted into methyl tetrahydrofolate, and this methyl tetrahydrofolate is responsible for homocysteine synthesis. Homocysteine synthesis. And this homocysteine is converted into methionine, and in this reaction there will be conversion of methyl THF into THF in presence of vitamin B12, right? So whenever there will be deficiency of the B12, it will lead into THF trap. THF trap, because THF won't be formed, and there will be abundant of methyl THF, and this abundant methyl THF and no THF is itself called as folate trap, right? And because there will be no conversion of homocysteine into methionine, it will lead to various issues such as various cardiovascular symptoms will be also appearing, right? So another thing is that this methyl THF, it gives TMP. It gives this methyl THF gives rise to TMP here, and this TMP is responsible for DNA synthesis. DNA synthesis, right? So this is how the Metabolism of one carbon compound is important, and methylene is the entry point. And the important thing that we have seen is in deficiency of vitamin B12. In deficiency of vitamin B12, there will be a folate trap, and this folate trap means there will be abundant of high THF, but there will be no THF. Right, now coming towards the deficiency manifestations of folic acid. So deficiency manifestations. So first one is megaloblastic anemia. So what happens in megaloblastic anemia is, the reason for megaloblastic anemia is in folate deficiency is because of the folic acid, which is responsible for DNA and RNA synthesis that we have just seen in last slide. And thus in deficiency there will be decreased synthesis of TMP. This TMP will be decreased synthesis of TMP, and that's why the C2 and C8 of purine will also be not formed, and there will be stoppage of cell division, and cells will be trapped in the S phase, and there will be very less cell growth, and there will be culmination of cell division, and thus the RBCs, they will increase in size. RBC increase in size, and there will be decreased size of nucleus. So that's why the word is coming from megaloblastic anemia, because of high amount of cytoplasm and little amount of nucleus. It looks macrocytic, looks macrocytic, and it is called as megaloblastic anemia. Right. So the second deficiency manifestation that we have to see is, as there is block in the conversion of homocysteine into methionine. This homocysteine would not be converted into methionine. So there will be increased level of homocysteine, and because of increased level of homocysteine, it will also cause homocysteinemia. Homocysteinemia. And because of this homocysteinemia, it will lead to various cardiovascular symptoms. Right? So this is the second deficiency manifestation. Coming to the third deficiency manifestation is neural tube defect. Is neural tube defect. So whenever there is decreased vitamin B9 in pregnancy, it will cause spina bifida. Right? So this is about the neural tube defect. Now, what are the causes? What are the actual causes of deficiency of folic acid? So, whenever there will be increased demand in pregnancy, increased demand in pregnancy, then because of various drug interactions such as phenytoin and phenobarbital, phenytoin and phenobarbital, right? Then because of the dietary deficiency, then because of various gastrointestinal surgeries, right? Then talking about the recommended daily allowance, RDA, that is 200 microgram normally and 400 microgram in women, pregnant women per day. Now, what are the sources? We have seen that green leafy vegetables are the good sources. So, spinach, beans, then orange, papaya, beet are some of the very good sources of folic acid. Now, talking about the antagonists of folate, antagonist of folate. So, it is used. So, these antagonists of folate, such as sulfonamide. So, the sulfonamide is actually antibacterial agent. Is antibacterial agent. So it has a structure which is quite similar to PAPA, and it inhibits the folic acid in bacteria, synthesis of folic acid in bacteria through competitive inhibition or competitive binding, and thus various kind of the antagonists of folate, they are used in treating the bacterial diseases or bacterial infections and various types of cancers as well. So the antagonists are used like this. Then the other antagonists of folate are aminopterin and uh, amethopterin. And this amethopterin is also called as methotrexate. So it is also used for treatment of cancer. So it inhibits the dihydrofolate, DHFR, dihydrofolate reductase. It inhibits DHFR, and actually these aminopterin and amethopterin, they are structurally similar to folic acid, and that is why they perform this competitive inhibition for dihydrofolate reductase, and there will be less synthesis of tetrahydrofolate, and this tetrahydrofolate is required for the proliferation of the cancerous cells, and when there will be binding of aminopterin and amethopterin, there will be no cell division, and because of the no cell divisions, the cancer cells will die, right? So these are this was about the antagonist of our folic acid. Now talking about the lab diagnosis of deficiency of folic acid. So first is peripheral smear. Peripheral smear. So this peripheral smear will actually tell about the macrocytic appearance of RBC, macrocytic RBC, and reticulocytes. Right. So first is peripheral smear. The second is histidine load test. Histidine load test. So when histidine is given, histidine is converted into figlu, that is formamino tetrahydrofolate, and this figlu is excreted in urine. So excretion of formamino uh, glutamic acid is indicative of deficiency of folic acid. So this figlu is formaminoglutamic acid. So whenever it is excreted out in urine, it is a clear indication of deficiency of folic acid, right? So whenever if there is tetrahydrofolate is there, if tetrahydrofolate is there, it uses the formamino group and it forms the formamino tetrahydrofolate, and thus figlu is not excreted in urine. So whenever there will be tetrahydrofolate, it will use up the formino group. And when there will be usage of formino group of forminoglutamic acid, there will be no formation of Figlu and no excretion of Figlu in urine, right? So this was about the second type of diagnosis. Now the third type of diagnosis is folate level in serum and RBC. So folate level in serum and RBC, right? So we have almost talked everything about folic acid. Let us move further and talk about vitamin B12. Vitamin B12, that is cobalamin. Just a second, just let me check whether I'm live or not. So just a second. Yeah, so I am clearly live. Let us just continue with vitamin B12. And so vitamin B12, that is cobalamin, and it is also called as hematopoietic vitamin, also called as hematopoietic vitamin. So what are important things about this cobalamin is it has got chlorine ring, then it has got cobalt in center, then it is also called as vitamin B12, and its active form is adenosine cobalamin, right? Then it requires intrinsic factor for absorption in ileum. It requires intrinsic factor for its absorption in ileum. Then another thing is there, if its a deficiency is there, it will cause various neurological manifestations. Then it is also called as extrinsic factor of Castle. Extrinsic factor of Castle. So these are some of the important points about vitamin B12, or that is cobalamin. So let us move further and talk about this vitamin in detail. So it catalyzes three reactions. It catalyzes three reactions. Catalyzes three reactions. So what are those reactions actually? It performs the methyl malonyl CoA mutase responsible for methyl malonyl CoA mutase. Then it is required in methionine synthesis. Methionine synthase. And whenever there will be deficiency of vitamin B12, what all it will lead into. So deficiency causes megaloblastic anemia, megaloblastic anemia, and it causes subacute combined degeneration of spinal cord. Subacute combined degeneration of spinal cord, and there will be excretion of methyl malonate in urine. There will be excretion of methyl malonate in urine, and it is also called as methyl malonic aciduria. Methyl malonic aciduria. Now talking about its digestion, its absorption and in transport in the body. So digestion, absorption and transport. So the salivary gland, it produces this R binder, heptocorin, heptocorin, or it is also called as cobalamin, cobalamin, right? So there are these parietal cells, gastric parietal cells as well, gastric parietal cells, and they secrete this intrinsic factor. So whenever there will be consumption of dietary vitamin B12 uh, is taken with the action of HCl and pepsin, there will be release of vitamin B12 in stomach whenever it is taken in the dietary form. So whenever this vitamin B12 is released, then it combines with R binder, this vitamin B12, and this thing occurs in the stomach. Right. So this is about stomach. Then talking about duodenum, what happens in duodenum? What will happen here? Transport. This will be transported to duodenum, where the protease action will cleave the R binder. R binder will be cleaved, and this vitamin B12 will join with with intrinsic factor. Will bind with intrinsic factor. Right. So whenever this will happen, the R binder and intrinsic factor, actually we have seen that very important for the digestion of vitamin B12 and its absorption. So when this intrinsic factor and vitamin B12, it will reach ileum. Now let us talk about ileum. What happens in ileum? Will reach ileum, there will be special receptors for the intrinsic factors, and these special receptors are called as tubulin, and these tubulins are present on the cells of ileum, or they are present on ileal cells. So when IF and B12 reaches tubulin with the help of endocytosis, this complex is internalized by the ileal mucosal cells, and then it to the portal circulation, goes to portal circulation, and it binds with transcobalamin 2. It binds with transcobalamin and 2. And like this, it reaches all the tissues and reaches different tissues. Right? So this is how it is absorbed, it is transported, and it is given to the various parts of the body. Now talking about the sources of the vitamin. So it is produced by the microorganisms of the intestinal flora also. So it is produced there. Then the good sources are meat and fish. Then talking about the RDA, recommended daily allowance. So it is like quite less. That is 1 microgram, and it itself is produced in the body. So not a big thing. Then talking about the biochemical functions of the vitamin, biochemical functions, biochemical functions. So it is required for two reactions. The first one is methionine synthase, and it is responsible for the methyl transfer reaction. So first place is homocysteine converted into methionine, and it requires vitamin B12. It requires vitamin B12, and there will be conversion of methyl tetrahydrofolate into tetrahydrofolate. So whenever there will be deficiency of vitamin B12, what will happen? There will be increased level of homocysteine, and it will lead to cardiovascular diseases, and there will be this folate trap. We have already talked about this folate trap previously, right? So this folate trap. So it is highly needed for active form of tetrahydrofolate. In second place where it is needed is conversion of methyl malonyl CoA into succinyl CoA right. So this is a vitamin B12 dependent reaction, and the enzyme is methyl malonyl CoA mutase. And why it is so important? Because it is coming from the fatty chain oxidation, that is from the propionyl OA. So that's why it is very important. So in vitamin B12 deficiency, methyl malonic acid is excreted in urine. This also we have seen clearly many times. Now talking about the deficiency of vitamin T12. So what are first of all the causes of its deficiency, that why the deficiency is actually occurring? So the deficiency reason is nutritional deficiency, malabsorption syndrome, then there is increased demand during pregnancy, increased demand in pregnancy, then pernicious anemia, that is the production of antibody against the parietal cell. Thus, there will be decreased intrinsic factor and there will be vitamin B12 deficiency. So, parietal cells, they themselves will be damaged. So, there will be, how will be the damage? By the production of antibodies. Now, talking about the symptoms of its deficiency. So, first is megaloblastic anemia. Megaloblastic anemia. So due to linking of vitamin B12 and folate in this reaction, there will be decreased DNA synthesis, and cells will be arrested in the S phase, and they'll continue to grow in size, and there will be lots of cytoplasm and very little amount of DNA, and they will appear macrocytic. So it is also called as macrocytic RBC, or there will be lots of immature RBC in the circulation, right? So this is about the megaloblastic anemia, and we have also seen subacute combined degeneration. Subacute combined degeneration. So due to defective myelination, it will cause defective myelination, and due to vitamin B12 deficiency, methyl malonyl CoA generation will be hampered. So this is how it occurs, and this methyl malonyl CoA, it gets incorporated in sphingolipids and glycolipids, and it will lead to defective myelination, and which is actually a degenerative process. So due to decreased vitamin B12, uh, there will be also excretion of methyl malonate in urine, right? So this all we have seen about vitamin B12, and I think I've tried to cover all the things which are related to vitamins, and I hope you people have understood what I was trying to actually convey to you all. So we'll be meeting in some other next class, and we'll be covering some other important topic of biochemistry. So take your dose of vitamins, go through all the lectures of vitamins. I've covered all the vitamins for now in the series of vitamin B. And as we say, let's crack NEET PG together. And don't forget to subscribe our channel, that is Let's Crack NEET PG. Also hit the bell icon for getting the notification for all the upcoming videos. And don't forget at all to use my referral code, that is FB01, for getting an additional 10% discount in your class subscription. And keep studying. Have a very good day. Thank you very much for your patient listening. Uh, Come back next time for the next video.